Hey everyone, today we're going to go over the sensation parts of all of your five senses, including going over some of the theories. So let's get started on the first one, which is vision. So vision, we are obviously have our vision just because we're using the physical energy of light for transduction. Color is not a property of the object, but sensation actually, it's the perception of the brain that creates that color. So color is determined by the wavelength of light as we look at properties of light and so forth, and reflects while it absorbs all the other colors. So black absorbs all wavelengths and reflects none, as you guys have learned in science, and white absorbs none and all wavelengths are reflected. A person has, within normal color vision, can see up to 120 or so colors. So three properties of light. We have hue, which is the variety of wavelength, and different wavelengths correspond to our, that we're subjected to different colors. Saturation or purity is of the light waves. So red is obviously really rich, while pink is sort of a combination of different wavelengths like red and white. And then brightness is that perceived intensity and the amplitude of those light waves. So those were a review from science that you guys had. It's important when looking at that because it's part of the sensation and perception. Now, as we look at the eye itself and how it works, I sort of broke this down using the parts of the eye, but also putting in blue the process in which the eye actually uses that. And we talk about vision and hearing and look at that more, as you can see, like there wasn't a lot on taste, because vision and hearing are what most people use to gather information about their world. Which is why we do an experiment on with Dharma and Greg on seeing and hearing. So first of all, receptor cells in the eye are sensitive to light. And we've talked about receptor cells and neurons and so forth. So that's why we build on that and get into different parts. And that way it's very easy for you to sort of transition into sensation. Light is going to pass through the cornea, the pupil, lenses, and that's going to be focused right on the retina. So when we look at the cornea and you look at the diagram I have there, the cornea is that clear membrane that helps to gather and direct the incoming light. The sclera is the white portion of your eye that covers your eyeball and has more of like a protective sort of substance part of it. The pupil is the black opening in the eye's center that changes size, and that's going to regulate the amount of light that's coming in to go into the retina. And the iris is actually what contracts and expands the pupil in order for it to do that. The lenses is that white disc that you see behind the iris, the color blue iris that's in the diagram. The lenses is going to actively focus or bend that light to fall onto the retina. And that process of light focusing and bending is called accommodation. So some of the problems that we can have with with vision, is because of the lenses. And if you look at the healthy eye and the astigmatized eye, the healthy eye has a sort of looks like almost like a smaller lenses. The lenses of the astigmatized eye looks a little bit bigger, and you can see the difference there. There's like the normal vision versus astigmatism, where you can see sort of the streaks of light, and that would really affect somebody's perception of what they're seeing and so forth, because functionally they're not able to see as well. Myopia is the idea of nearsightedness. So distant objects look blurry, but you can see near, so you are nearsighted. And that's due to the light reflecting off objects in front of the retina. Hyperopia is the farsightedness, and that's objects that look blurry. Close are going to be blurry, and then far away you can see, and that's because light is reflected off of objects and it's focused behind the retina. Two other things in terms of, you can see in terms of age, is the brittleness and inflexibility of lenses, and then of course, as we talked about, the curved eyeball or lenses, which can create blurriness in a particular direction. These can be fixed in a couple of ways. Eyeglasses help to and so does LASIK, which reshapes the cornea and so forth, help to bend the light correctly so it falls on the retina correctly. And eyeglasses would do that as well, and contacts. So as we continue, so the light is on, striking the retina in the back of the eye, and because of that, that activates, that physical energy is going to activate the rods and cones, which are known as your sensory receptors or your photoreceptors. So when those are exposed to light, they have a chemical reaction that's going to create that neural signal that we've been talking about with neurotransmission. Rods are your thin, long ones, so you'll see here on the side. They're sort of that gray color, and they're gray because they are not sensitive to color, while you see the cones are, blue, red, and green. They are not sensitive to color, and we use them mostly when we are getting up in the middle of the night and we need to find our way through a dark room. They can detect visual stimuli in the dark much better than a cone can. They do adapt slowly to changes, so it does take about 30 minutes to reach the maximum sensitivity, and they are located in the peripheral of the retina or the sides. The cones are much shorter and fatter. We have less of them in the body or in the eye, and they detect color and are responsible for visual acuity because of the type of messages that go to them. So all that fine details that we look at things, that makes sense because we don't find a lot of fine details in the middle of the dark, which is when our rods are working. They do adapt quickly, so that's why it's much more easier to adapt to bright light than it is for dark. It takes a little longer to adapt to the dark and so forth, so it takes about 5 minutes to adapt to bright light. It requires more light to distinguish color, and that's one of the reasons why cones are susceptible to bright light. And they are located in the fovea, which is in the center of the retina. And we'll do a test of that to show you where your rods and cones are when you come back into the classroom. But there's less of them as you go away from the center of the retina. So in the peripheral, we have more of the rods. The optical disc is the only place that doesn't have rods and cones, and it's known as our blind spot, where we can't see that. Okay, so after the rods and cones are signaled, there's a collection by the bipolar cells. And so you can see in this diagram, the rods and the cones are on the bottom there. And then you'll see the bipolar cells, or the specialized neurons, and then those are connected to the ganglion cells. So basically, it's just rods and cones are going to, those signals are going to be collected by the bipolar and then transferred and transmitted to the ganglion cells, which then go to the optic nerve. Each ganglion cell receives information from photoreceptors in a receptive field in the retina, and then the ganglion cells are going to combine, analyze, and encode that information so it can be brought to the brain. Signals from rods and cones are definitely processed differently. We get information from 1 or 2 cones, but 100 or more from rods. So the rod information is combined before it goes to the brain because there's just so much going on. Ganglion cell axons form the optic nerve, so you can even see that. There's the same color as the optic nerve, okay? And that's going to directly transmit information to the brain. So less specific visual information is sent from rods because there's so many of them, and there's more visual detail from the 1 or 2 cones, and that makes it so the ability for more visual acuity or detail is much better with cones. Okay, and then the last things that happen before it goes to the brain is that the left and right optic nerves are going to meet at the optic chiasm. And I have this diagram here so you can see what it looks like. So the optic nerves are coming from each of the eyeballs, and then the optic chiasm you can see is that sort of crisscross that they have where the fibers are going to cross to the opposite sides to the brain. And so that makes sense that when we're looking at visually, the right is the left and the left is the right, and it's doing that quadrilateral organization. So when it processes by the brain, most of those optic nerve axons are going to travel to the thalamus in the limbic system, which is going to process everything to see what's going on. It's a sensory processor for form, shape, brightness, and depth. And then some of those are going to go to the midbrain before the thalamus because it's going to process the location of the object. Signals are then sent to the visual cortex, which you can see on the diagrams towards the back in the occipital lobe, where they're going to be decoded and interpreted. And there's feature detectors there, so parts of the visual cortex are going to specialize in visual stimulus, and it's a very complicated part of the brain where it's just like, okay, well, where is it in the visual cortex? So there's just certain detectors. That's what makes it amazing about our brain, that just look at angles, lines, movement, and so forth, so it can help you interpret that. Then those things are going to go to the frontal lobe and other regions that recognize the image, use it, if it's a pattern, it's something that you remember seeing, and so forth. Color and motion perception are developed in infancy. And so those things are pretty established in us as we get older. But new in objects and faces have been shown to have plasticity because those things evolve over time, and we have lots of things that we learn that are new. But color and motion are something that's really fixed at infancy. Okay, and then vision theories. In terms of rods and cones. Like I said, we're going to do the shake color pen test when we get into class. I'll be going sort of back over just a little bit, not much, but a little bit, just to do some experiments with you. So rods and cones. So rods are in the peripheral vision, as we said, and the cones are in the fovea and scattered throughout the retina. And so we'll do that pen shake test. But one of the visions of theories is the chromatic theory. And that is that we have 3 varieties of cones. Each cone is sensitive to a certain wavelength. So red sensitivity is long, green sensitivity is medium, and blue is short waves. And that other colors are stimulated with a combination of cones, like a color wheel. So you'd have a combination of these 3. But the chromatic theory, that's why you saw red, green, and blue on the diagram, because of this chromatic theory. Remember, vision theories are all correct. It's just a kind of a combination in what each one kind of talks about. And so we have these 3 colors that we are sensitive in terms of that, and then after that it's kind of combined. In terms of application of this theory, we can kind of look at it when we look at color blindness. So if somebody is red-green colorblind, which is the most common, okay, that's when you have normally blue sensitive cones, but the others are either red or green. You don't have both. So red and green look the same. You can distinguish blue because that's a sensitive cone, but either red or green is going to look the same. And that's why they put a stoplight vertical, so that people that are red-green colorblind, they are not going to get confused. They still see, they know that red's on the top and green is at the bottom. And so that way they are still safe on the road. And if they see the top one lit up, they know it's a red, and so forth, and they can still tell that. And that's why they are that way, and it's the most common color blindness. The opponent process theory proposes that there's 4 basic colors divided into 2 pairs of colors, and this one includes yellow. So yellow is included in that. And you notice that they're all primary colors that we learn about in art. So this one says that there's 2 opponents. So green and red go together, and blue and yellow. So if red is stimulated, then green is inhibited. And if green is stimulated, then red is inhibited, but they go together. Black and white are going to act as opposite pairs as well. And color is sensed and encoded by the proportion of red and green or blue and yellow. And so that's how it's determined whether or not that red is stimulated or green is stimulated. The other pairs, other colors stimulate one of the pairs. So purple stimulates red in the red-green pair, plus blue in the blue-green and the blue-yellow pair. So this one is more interesting because I think this one can make a lot of sense to people, because we have more than red, green, and blue colors, as the trichromatic theory would state. But you can also then prove, color blindness, that with red and green in the opposite pairs, one isn't working. One is working, they look the same. So the opponent processes theory really brings in, if somebody's functionally working, that red and green would still be able to be either stimulated or inhibited. But purple is interesting because purple isn't part of that, basic 4 colors. But again, it goes with when we combine colors in art, that it kind of goes together. So just remember that red and green go together, and blue and yellow, and it depends on the proportion of that color of which one's going to come out. So let's try this, and we'll try it again in class, but let's try this. And my face may throw it off, but let's just try it. And like I said, we'll do it in class to see, and we'll see if it works. But this is the opponent process theory at its best. So what I want you to do is stare at the middle white dot, okay? And you're going to stare for 30 seconds. Keep staring at it. Don't look down. Just keep staring at it. Keep staring at it. Don't get distracted and look away. Don't keep distracted. Now, what you should have seen is a regular flag, a red, white, and blue flag, which is pretty cool. So when you're staring at that red and that green and yellow flag, you're using your cones for yellow and green. And so this is this after image phenomena that you read about in your book is really true, because it shows that once those are exhausted, then the other pairs are going to show up, which is why you get blue and red, which is pretty cool, which is the opposite pairs, obviously. So when we stimulate those, we exhaust them, and then the other pairs kind of pick up, and that's where we get that temporary after image. So again, if it didn't work for you, we'll do it again in class, see what it looks like, but I think it worked for me as too. Okay, so here what you see is color deficiency. Like, I'm sure all of you have seen this at a doctor's office. You know, the red and the green. They're trying to see if you're colorblind, because they're trying to look at those cones and see if you're actually seeing them both the same color, or you're actually seeing them different. So that's what you'll see: blue and yellow, and also green and red. Okay, let's go into hearing. So hearing is really important for us for survival, social interaction, and language development. And there's some characteristics of hearing. So loudness is the intensity or amplitude of the sound waves, or they're called decibels. So 0 decibels is an absolute threshold and the softest sound. We also have pitch, which is the highest or lowest of a sound, determined by the frequency of those sound waves or rate of vibration. So the number of peaks per second, so you know, something that has a lower one's going to have, or less vibration, has more space in between those wavelengths, and then you know, something higher is going to be a lot shorter wave distance between those wave peaks. Most sounds are very complex. They are consistent of several wave frequencies, which produce a unique quality to the sound called timbre. So if you play E on a guitar, you can hear the same note on a saxophone or piano, because that timbre is very unique to that, playing that note E. Okay, the process of hearing, like I said, this is the other one that's you know, really detailed, because is one of the ways that we do process information. The process of hearing includes the outer ear, include the pinna, the ear canal, and the eardrum. You can see there on the diagram, the pinna is this part of your ear, just this, you know, flappy part. It's all going to collect and channel those sound waves into the ear canal, which is that long one there. And then it hits the eardrum. So the sound waves travels down, and it's going to vibrate depending on the intensity and frequency of that sound wave. That then goes to the middle ear, which is the hammer, anvil, and the stirrup. Those also have different names as well. As you can see in the bottom diagram, those are the proper names for them. The eardrum vibrates. When that vibrates, it transfers into these 3 little tiny bones, and those are set in motion, and that's going to amplify the sound. The stirrup then transmits that vibration to the oval window, okay, and that is a membrane that's going to transfer the vibration to the inner ear. And the inner ear is your cochlea and your semicircular canals. The cochlea is that snail-looking fluid-filled tube that includes the basilar membrane and also your hair cells. And then the semicircular tubes that you see are those kind of just above it. And those semicircular tubes has to do with your vestibular sense. And if that's off a little bit, the fluid's off, and then you're going to feel a bit tipsy and a little off balance because the fluid in your ears. If any of you have had vertigo, those things would have been affected. So in the cochlear shell, okay, or the shell, I shouldn't say shell, but it's like that, there is a liner called the basal membrane. You can see that, and to the right of the basilar membrane diagram I have here is the semicircular canals. But that cochlea is lined with the basilar membrane, and then on the basilar membrane are all of the hair cells, okay? And when the vibration comes through the oval window, it's then going to vibrate the fluid in the cochlea. And once that vibrates, it's going to transmit that to the basilar membrane inside, and then the hair cells are actually going to start to bend and convert that into a neural impulse. Okay, and that's going to stimulate the cells then of the auditory nerve, which is going to carry that information to the thalamus and then the auditory cortex, just like it does with vision. Okay, and then hearing theories for vision, we're going to do a couple things in class. As you can see, there's some videos there. I'm going to be showing those in class, but distinguishing pitch, the basilar membrane is important for this. The frequency theory says that the basilar membrane vibrates as the same frequency as the sound waves coming in. Okay, so if there's 100 hertz, then the vibration is 100 times per second. There is a limit to how fast neurons can fire, okay? And so it can't fire faster than 100 times per second, okay? One of the things we're going to do in class, though, is we're going to do an experiment. I'm much older than you, and you're younger, obviously, and there is a difference between what adults can hear and what children can hear. And so we're going to do a little experiment with the frequency theory. But this has to do with the vibration being the same as the frequency, but that you do have a limit to how fast you can go. Can't hear everything. That's why sometimes really, really high pitches, not anybody can hear, and dogs can hear it, so they have different capacity than we do. Okay, the other theory is the volley theory, and there you can see a hair cell. This is a hair cell from a frog, but ours looks very similar. And again, those are inside the cochlea, and this is an explanation for higher pitch. You know, how we can actually hear a higher pitch. So the volley theory says that the hair cells are going to fire like volleys, and you can imagine like when they used to do wars and line up like Napoleon style and just shoot and have to reload and so forth. People could die in between because having to reload and just shoot everything all at once and reload, you could be susceptible to crossfire or fire from the enemy. So this one is like, let's have this section fire, then that section fire, then that section fire. By the time this one is done, this one has done, this one has reloaded, and we can keep doing that. So this is so neurons take turns instead of firing in unison, and impulses are going to be sent to the brain at higher rates, and that would explain why we can hear pitch much higher as opposed to the frequency theory that says that there's some limit to it. Okay, they both are very correct. It's just that one talks about the frequency matching the sound waves, and this one's going to extend that one and say, okay, this is why we can hear things that are a much higher pitch. Okay, the last one is place theory, and that's different frequencies cause larger vibrations at different locations. So again, the idea of place, the place in which it is processed and so forth. So high frequency sounds, the maximum vibration is near the stirrup end of the basilar membrane. The higher frequency is interpreted in the place where where the hair cells are the most active. So when hair cells are much more active, obviously then the activity and the sensation or the sensory input is going to be much higher, and so there can be higher frequency sounds. So different pitches are going to excite different hair cells as well. And so lower frequency sounds, the maximum vibration is at the opposite end of the basilar membrane, and so that's responsible for different types of hearing as we look at where it's actually processed, okay, and the different vibrations that occur. Okay. All right, for hearing loss, this is really important. Here's you can see some hair cells of humans at the top here, and that's where we want to talk about conduction, deafness, and also conductive. As we get older and listen to our music in our ears and our earbuds, we need to be really careful because we can damage permanently our hair cells. And if you damage your hair cells, they do not grow back. Okay, it's not a part of your brain. There's no plasticity or neurogenesis, and so you have to be really careful of how loud you have your music in your car, and especially having earbuds that are really close. If you can hear somebody else's music, then it's too loud. Okay. So the different types of ear damage, some of which people are born with, but conduction deafness is the tiny bones, all those 3 bones in the middle ear are damaged or become brittle with old age. And you can fix this with a hearing aid, okay? Conductive is limited, is there is a limited motion in the ear. So any of the things that are put in motion, like those three bones in the middle ear, that is the limited motion. It's not the brittleness of them, it's the actual motion is slowed down, so the hearing is actually reduced, and that can be also fixed with a hearing aid. Then there's sensory neural or nerve damage, and that's damage to the cochlea, hair cells, like we just talked about, and auditory nerves. And so if you want to fix this, it has to be a cochlear implant, because if we damage our hair cells with our earbuds, then we are taking away the apparatus that helps with neural signaling. And that is when you need a cochlear implant and so forth. If someone's born with a damaged cochlea or born with some kind of neurological issue with their auditory nerve, then they'll have that. And that device changes the sound waves into an electrical signal. So it kind of takes the place of the cochlea or the hair cells or the nerve, whatever's damaged. Tinnitus is a ringing or noise in the ear. Some of us get that. And this one can be a sign of an underlying condition. I've heard things with the heart problems with the ringing in the ears. It can also be caused by age or ear injury and so forth. 1 in 4 by 2050 will lose their hearing due to damage we caused by ourselves. So the rule is a 60-60 rule. Only have earbuds up 60% of the maximum you go on a device and no longer than 60 minutes, which I know when you're doing things online, it's not really helpful. So at least keep it down. And remember that sensory adaptation is going to take over for you. So when you have those earbuds in and you've had them in for a while, and then you're going to be adapting and you're going to keep turning the volume up, okay? When you take it out and put it back in after a while at the same volume, you're going to find that you need to turn it down. And I know I've done the same thing where you just get used to the sound and that can really damage the ears. So be careful with your ears and your earbuds. Okay, this was what a cochlear implant looks like. If you can look on the diagram, you can see people they have something attached to the outside of their head, like you see the little girl there. There's a sound processor and a transmitter and a receiver, and you can see it goes right into the cochlea, which is going to help with getting that signal going, okay? So a microphone picks up the sound from the environment, and then it's going to convert it into an electrical impulse and stimulate the auditory nerve and so forth. Okay, taste and smell are definitely connected. We often, if we lose our sense of smell when we have a cold, we often can't taste anything. When we inhale the air molecules from a substance and that's going to go back to the back of our throat. And the air molecules and chemicals are what we use for smell and taste as well. We can tell the difference between honey and candy at the same time due to the connection of smell. So smell when you are tasting something can often help with determining. So they work hand in hand. Sensory adaptation is reached in less than 1 minute with 70% less sensitivity to it, which is pretty amazing. We just kind of get used to something. Think about if you eat a block of cheese and you have just cheese. There's nothing, there's no crackers, there's no jelly on it, there's nothing. It's just cheese. How much you kind of get used to the taste of it and almost get sick of it. And it doesn't take very long to taste something with just having that one taste before you do get sick of it. One thing to keep in mind is that they have all these diagrams all over the internet about tongue maps and things like that. They have certain spots. Your entire tongue takes up all those different tastes, and so that is sort of a fallacy. Okay, so part of taste and smell and the brain is the different parts. The one picture I have at the beginning is actually really good. The parietal lobe is there to process information about temperature and taste. And if we're touching food and so forth, the occipital lobe is the visual portion of that, and the temporal lobe integrates all of that in order for us to help remember. Also the auditory parts of food, crunching, slurping, those kinds of things are all used in those parts of the brain. And smell really matters. How you look at smell. Out of the five senses, smell is mostly closely linked to pleasant memories. Smell plays a role in overall sensory experience as we either enhance it or lessen it. After smelling a strong scent, nerve cells begin to fire less frequently, causing strong smell to be less distracting for some. And it's like when you put on cologne or perfume for the first time and you can really, really smell it, and then after a while you just don't smell it anymore. So sensory adaptation really takes hold. When you're sitting in Chili's, if you've ever been in Chili's, and then you come home and you can smell your clothes, especially if you put them down and then pick them up the next day. But because you're at Chili's and you're kind of engulfed in all those smells, like that sensory adaptation takes over. And so it's kind of interesting to see how smell is affected by that. So how does smell occur? Of course, smell is responding to chemical substances. It's really important for protection because if we have spoiled food or a gas leak, it is for our protection. So it's innately created that way. The sensory stimuli that are used are the environmental energy is molecules in the air that are inhaled into the nose or in the opening in the back of the palate of the back of the throat. And in the nose, the molecules, first off, have come in contact with millions of olfactory receptor nerves. And you can see in the top example or diagram, you can see the olfactory receptors there as that McDonald's or food, pizza, whatever comes in. And it's the top of the nasal cavity there, you'll see those receptor cells. And they're constantly being replaced. 30 to 60 days is when they actually last. And each of those receptor cells responds to a specific chemical structure that will help to determine what it is. The stimulation of those receptor cells is going to then create the neural passage or message along the axons to the olfactory nerve. And you can see in the bottom portions just above my head here, the olfactory nerve, just below those are the receptors, and just above that, above the olfactory bulb, is the actual nerve, and that's where the neural messages are being sent. Then the molecules activate specific combinations of receptors that allow the brain to interpret a pattern, which is pretty cool because as patterns happen, then you can identify what an odor is. So even if you were to close your eyes, because you have identified over time, or even the receptors, because they are working in conjunction with each other, that they create a pattern, and then you can specify what an odor is. The olfactory nerve is then connects to the olfactory bulb, and you can really see it in this diagram here. You've got the receptor there at the bottom on the left-hand side, and then you see the synapses, like there's even spaces. You can even see some of those in those cells that you can see in the olfactory bulb. So you really see like the like the neurons and the ideas that we learned from last chapter there, and those are a part of the olfactory bulb, and then that's going to go out and up to the brain. So the olfactory nerves connect to the olfactory bulb and the enlarged end of that olfactory cortex at the front of your brain. And what's really cool is your brain directly, it doesn't go through the thalamus, it goes directly to the brain. And so there's this direct passageway to the brain from your actual nose. Axons from the bulb form the olfactory tract, which then connects to the temporal lobe and the limbic system immediately, so you can recognize those smells. And then the limbic system will put an emotional response to smell as well. So how do you have smell reduction? Well, it does decline over age, which is really interesting. If you watch an older person, I know my grandmother does this, 50% of those 65 and older, it declines. And if you think about it, they eat just about anything. So Brussels sprouts don't bother them anymore because they're not as strong tasting or smelling. They eat way more butter and salt. So those are a lot of things in terms of health issues. And my grandmother used to do that all the time. She would just pound on the salt and the butter. And it's might be because they can't smell it as well. Also, air pollution, smoking, and exposure to any kind of chemicals would decrease stability. Watching out for cleaning products. So I'm big on plant-based cleaning products because of that. We have enough air pollution, especially in Indiana. Indiana is huge for air pollution. in fact, and that's why our allergies are so bad. Of course, you can help smoking or being around those and trying to have those around you who are smoking to reduce it, but also using different chemical products in your home that are safe and are more plant-based and non-toxic. That's really important. There's also a loss of function associated with Parkinson's disease, schizophrenia, and multiple sclerosis, as it's stated in your book, and possibly Alzheimer's. So why is it that happens? I know your book doesn't talk about that. So Parkinson's actually is correlated from a buildup, a clump of protein. That's the hallmark of Parkinson's. And it may form in these parts of the bodies first, where you have smell, and so they think that protein builds up there. In schizophrenics, they lose brain volume, and particularly they found in olfactory bulbs, interestingly enough, schizophrenic people lose that brain volume there first. And then those with multiple sclerosis or MS can lose the ability to distinguish all of the four taste qualities, and that's a signal associated because of lesions on the brain that would create that problem. And then finally, there was a section in your book about pheromones and human chemical signals. So animals use pheromones to communicate social, sexual status, marking territory, and for warning signs. And Martha McClintock in the University of Chicago found that some women in dorms or people in the same household would also see this, form and develop the same menstrual cycles. So their period happens, and that she thought was related to underarm sweat that is being sort of distributed around the dorm room. And this is called the McClintock effect. You definitely need to know that word, McClintock effect. So what they found is that human chemical signals found in steroids are located in sweat, armpit, blood, and semen. And so they're involved in certain emotional states, stress, anxiety, and fear. And some of those that were found by another researcher, de Groot, he was in 2012, because McClintock's was been, a bit earlier than that, 1971. It found a similar kind of connection that men watched a scary film or a disgusting film, and then they collected their disgusting sweat that they produced while watching those films. The women watched in this study watched a neutral film, and then when they were exposed to this sweat from the men, which I know is so gross, the women actually produced disgusting facial expressions even with the neutral film because these signals were making them feel an emotional state. Now, even though those things are somewhat true, there's no scientific evidence that any pheromone can be put in a spray, a pill. There can't be any like love potion or anything like that to make people feel a certain way. But certainly there's some kind of signal there that we've found, human chemical signals that can be found to influence people from another person. Okay, let's talk about taste. Taste also, like smell, protects us. Sweets attract us. Sweet tastes attract us to energy-rich food like oranges. Edamame is protein-rich foods like meat and proteins and cheeses. Bitter or sour warns us that there might be some kind of danger, and sensitivity to salt really helps us regulate our electrolytes. So the stimulus for this, again, is chemical substances. It's just like smell, except those are molecules, of course, and these are chemical substances. And what happens first is that the substances are dissolved by saliva, and the chemicals activate the taste buds. And each of those taste buds have 50 receptor cells that are specialized for a taste. And so at the top, you can see the taste receptor cell, and sort of the opening to the tongue. And each taste bud is going to show a maximum sensitivity to one particular taste and then less sensitivity to other tastes. But unlike where people say you have a sour section and a salty section, it still has some sensitivity, there we go, to other tastes. So we wouldn't say that it's just one or the other, but there are particular taste buds that go with one or the other, one taste or the other. When that happens, it's going to activate special receptor cells in the taste buds, and that is going to send a neural message. It's pretty simple. Dissolving, it's going to activate the taste buds. It's going to activate those special receptor cells. That's going to then send a message along pathways to the thalamus, and then the thalamus is going to direct it into several regions into the cortex. And you can kind of see that. I know that diagram's kind of covered by my face, but kind of eating in there, and then you'll see sort of the pathway up through the thalamus to maybe even the frontal cortex and so forth, is that direct neural pathway. Okay, and then lastly we have touch. We have touch in terms of pressure. So the fact that if I can feel somebody's hand on my shoulder. Obviously pain is a part of that, cold and hot, due to sensory receptors that we have around our body. And the skin, as it said in your book, is the largest organ that you have, 20 square feet and 60 pounds. The main part of it is Pacinian corpus, and that's a picture of it right there. So the Pacinian corpus is here, and that's how you see it under a microscope. And then if you look to the very top of the diagram, you'll see little bulbs. They're kind of in the dermis, I guess, technically. Those are where you're going to feel, it's kind of like a cushion, and those little bulbs will feel some pressure. So under the skin, and they are stimulated by pressure. And when they are stimulated, that's going to convert, the stimulation is going to convert into a neural message that's going to be relayed to the brain. If it has constant pressure, you're going to have sensory adaptation that's going to take place. So and that's going to happen because it's either your brain's either reducing the number of signals, or it's going to stop the signals altogether. So the interesting part is, like when you put your clothing on, you know what it feels like when you're putting on like a nice warm sweatshirt or some kind of like new sweater, maybe that's a little heavier, a light t-shirt, tank top, things like that. Like it has a certain feel on your body, but after a while you don't feel like you have it on, right? You can feel that comfort at first, and then that kind of goes away as the day goes on, and you don't really notice necessarily that you have anything weighted on you, okay? Pain definitely, like all the other senses, is for survival, and it's going to warn you for potential. It's going to have those withdrawal reflexes. We talked about spinal reflexes, so that your body does protect itself when it has pain. And there are some conditions where people do not feel a lot of pain, and so they hurt themselves quite often, and that can be problematic to the body. Okay, so for pain, we have, obviously pain is unpleasant sensory and emotional experiences that are associated with acute or potential tissue damage. And pain is a major part of this section, and it has a lot of things with it. Of course, with pressure, we talked about Pacinian corpus like taking on the pressure and the neural messages, and that's kind of normal. Pain has a whole other regulation and set of theories. So with pain, our pain receptors are called nociceptors, or they're also called free nerve endings. And so in this diagram, you're going to see some receptors for cold and for light touching and for heat, and then you'll see the ones for pain, and they're really up close, and they do have these kind of like, almost look like little dendrite sections. If you follow that all the way down, then you're going to see that nerve that's connected to it. And so those nociceptors are the free endings in the skin. And our fingertips, when you cut your fingertip, I know we've talked about that before, this has 1,200 nociceptors per square inch. Muscles and joints have fewer, which means that we don't feel as much pain, and organs have the smallest number of nociceptors. So what's interesting is that you could feel pain because you have something going on with an organ, but it's probably something else around it that's feeling, that actually has receptors that is feeling it, because our organs have less than fingertips even do. Hands, face, and lips are densely packed with those. That's why when you cut them or hurt them, they hurt the most, okay? And then what happens when an injury occurs? There's two types of nociceptors that actually are activated. One is an A delta fiber, and another is a C fiber. There's also an alpha beta fiber, but that one is a non-painful stimulus. So over to the right, kind of near my head, is what the skin looks like. The first one, that's 30 to 70 ms, is an alpha beta. That means that this is the threshold of pain. It has a much higher threshold of pain. And then your your C fibers are over to the right, and they have like less of a threshold. And then your A delta, I was trying to say it right. Your A delta fibers are the ones in the middle that have a threshold that is related to their fast pain system. So the A delta fibers are what you're going to feel first, like that initial, like really sharp pain, and those were myelinated, and that's because it's going to be this sharp pain that's going to help you protect yourself. If it was unmyelinated, you might end up slicing all of your fingertip off instead of just the beginning, because you need that quick, sharp pain to come quickly so that you do protect yourself. It is short-lived, and it is because of the immediate injury. The C fibers are then represent the slow pain system. So that's like right after that, the throbbing, the burning, the stuff that really hurts until it heals. And those are unmyelinated, so they come a little bit slower. Both fibers transmit a message to the spinal cord, which is going to show, do a reflex, but then it's going to cross to the other side of the brain. So then we get that ascending nerve signal. Most C fibers produce a pain enhancer called substance P, which is a neurotransmitter, and it stimulates the nerve endings at the injury and increase pain messages to the spinal cord. We don't want this. So if there's ways that we can help psychologically to reduce pain, we can do that. And so pain factors or psychological factors, the way that you have a relationship with pain, some of us have more pain tolerance than others, as they would say. If you're under stress, then pain could be a little bit more painful, because substance P is not going to be counteracted like with our natural painkillers, like endorphins and acetylphenides. And so those two could actually inhibit substance P and reduce pain if they are. But if you have something psychological going on, then those two natural painkillers will not be released, and you'll feel more pain. So after those are initiated, the alpha delta and then the C fibers, the fast pain messages are going to travel through the thalamus, as we said. It's all that sensory messaging. And then it's going to go to the somatosensory cortex, and that's where it's going to interpret the pain, its location and intensity. The slow pain messages then travel from this, that's fast pain. The slow pain messages travel from the spinal cord and to the hypothalamus, which is going to sort of regulate like our survival mode. It's going to go obviously to the thalamus because it's going to do that sensory, and then the amygdala, and that's where we're going to have that emotional tie. If it's something that's going to be slow and painful and last a long time, then we have sort of this relationship with pain, and we're going to remember the last time we did XYZ, we may not do it again because it was so painful. So it creates that memory and that emotion, okay? Morphine and opioids that are used for medication work on slow pain systems only. They wouldn't work on a fast pain. It's just too quick. So morphine works on things with slow pain. And then theories of pain. This is really interesting. Think about how you relate to this yourself. So we integrate situational information and psychological. The gate control theory is really cool. It depends on how the brain interprets pain, and against that pain tolerance. It regulates pain by sending signals from the spinal cord, and you either have an open or a closed pain gate. If you have open gates, and meaning you have this relationship and experience with pain that you just don't like pain, and it gets you upset more. I know my daughter's exactly like that. Everything is so painful and intense. She's got open gates. Mine's more closed gates. I can deal with pain a lot better than she can, so my pain is more reduced. And even if you were to try to calm yourself during a painful situation, you could even close those gates a little bit better. So it kind of makes sense that that's happening. And then when we think about again, our effects of perception on pain, we have to think about how our anxiety, our fear, feeling helpless in a situation, do you feel like you're out of control, you have no control over the situation. in depression and sadness and mood can actually affect how you have a relationship with pain, and also cultural beliefs and social norms. So the cultural beliefs of your, country or of your locality can have an idea of like what's an appropriate response for pain. Remember social norms of gender too. Do men cry when they're in pain and women do? And that's sort of like this social norm and stereotypical, like, women always cry, they're, when they're in pain. Or is your social norms in your country or culture that men, it's okay to cry? And so that can have an effect on how people show that they're under pain. Um, and then it could be in a family as well. It could be that the culture accepts pain and showing that you are under pain, but like your specific family feels like, that certain genders or you yourself, we just don't respond to pain and so forth. So when we come back into class, we're going to look at a case study in the Amazon about pain. And we think about that, athletes, as the person above you can see in the picture, condition themselves to minimize pain during competition. I know this happened this week. My daughter rolled her ankle in a soccer game, and she played the entire game, and then afterwards she was in so much pain, and it was because her endorphins were running at that time. It was really reducing the pain at the time, so she could run through it. Even though she could still feel the pain, it wasn't as bad as when she stopped. And those endorphins really helped that. But she pushed herself to finish the game because she rolled it at the beginning.